So this is the second of two discussions about the digestive system. This presentation will pick up from where we left off before. In the first discussion, we were starting with the oral cavity, of course, going through the pharynx, the esophagus, the stomach, and the small intestine, and also describes the large intestine. Along the way, we learned about the histology, what cell types and what special characteristics are found along the way. Most of the anatomy was left for you to review. Also discuss some hormones important in the digestive process. Go back and look at gastrin in the stomach and secretin and CCK. Know which digestive products are going to upregulate or cause more synthesis of those particular hormones, and also just appreciate the overall processes that are going through the digestive system. Now, today, I'll be picking up at 22.17. We've been through the colon, described the parts of the colon, and just the last little bit about the true digestive organs, and then we'll move over to the accessory organs of digestion. So when you look at the large intestinal wall from just a gross standpoint, you would notice there's no villi. There are none of these finger-like extensions moving into the mucosa, because we've already done the majority of we've already done the absorption of the nutrients, and of course all we need to do now is absorb the water and some vitamins from the bacteria. There are, however, some distinctive glands found in the large intestine, and these are going to be mucus cells, and it makes sense that you would want a nice, moist, lubricated colon. The mucosa here does not produce any enzymes. Again, all of the digestive processes as far as breaking down and absorbing nutrients has already been done previous to this. So what you'll notice is that we're back to a simple columnar epithelium. There are some of these intestinal glands along the way with lots of mucus producing cells. There's still what we're expecting, the submucosal layer, the muscularis, that hasn't changed. And then of course, we have on the outside a serosa or an adventitia. It is serosa if it is surrounded by the serous membrane. Recall that the ascending and the descending colon are retroperitoneal, so that outer layer, rather than being called a serosa, would instead be called an adventitia. Again, it's just a change in name because there is no serous membrane there because it's retroperitoneal. So we change the outer layer to the name of an adventitia. Following from the rectum, the anal canal. Again, there's going to be some longitudinal folds in here, some anal columns. We're going to move back from a simple columnar epithelium now to a stratified squamous epithelium. Now we're thinking about protection again. There's going to be a large number of veins running through that lamina propria and the submucosa, and an increase in this venous pressure is what can distend those veins, leading to hemorrhoids. That pressure is increased during defecation and also during pregnancy. So one complaint of women during pregnancy is hemorrhoids. There are two sphincters as you get to the anus. There's an internal anal and an external anal sphincter. The first one, the internal anal sphincter, is going to be smooth muscle, not under voluntary control. The second one is skeletal muscle, under which you hopefully have voluntary control. And then the anus, the very end of the anal canal, does have stratified squamous epithelium here that is keratinized. So looking at a cartoon representation of this, here is the last of the sigmoidal colon, and then the rectum, the straight region. As we look at it, we can see these anal columns and the two sphincters. Here's the internal sphincter and then the external sphincter with the anus itself. Now again, we're really not in the business here of absorbing nutrients from the diet, but so only a small percentage of the nutrients are going to be absorbed here. What is importantly absorbed here, however, are biotin, vitamin K, and vitamin B5. And these are all very important vitamins that are made in part by the bacteria in the colon. So the bacteria are there, they're producing these very important vitamins. There's also the important job of water resorption. This is going to help prevent dehydration. And while about 1,500 mils of material is going to enter the colon, over a liter of that will be reabsorbed as water through osmosis, and the remaining 200 mils or so will be released as feces. Of the 200 mils in the feces, it's still largely water, and then the rest of the feces are the indigestible materials, the epithelial cells that are traveling along, and about 5% of the feces are bacteria. This is what gives you the odor. There's some ammonia in there. There's some nitrogen-containing compounds. There is some hydrogen sulfide giving you that rotten egg odor. So again, what we see is each section is a haustrum. So you can see here in the colon, the major responsibility is the absorption of the vitamins as well as water reabsorption, with only about 200 mils or so ejected in the feces. There is a reflex that's responsible for defecation. This is going to begin with the distension of the rectal wall, triggered by some stretch receptors within the wall. And there are two positive feedback loops. Remember, positive feedback loops are those situations where once begun, they go to completion. There's a long reflex. This is going to be coordinated by your sacral parasympathetic system. This is going to be the mass movement of feces pushing toward the rectum. And then there's going to be a short reflex stimulated by those myenteric plexus that you see in the colon and the rectum. This is going to cause relaxation of the internal sphincter and moving feces into the anal canal, and then voluntary relaxation of the external sphincter is necessary. So this diagram from your book, 22.17, is showing you the basic positive feedback loop that is involved, and refers back also to the parasympathetic control that is occurring. So take a look at that. Just think that's controlled in part by your spinal cord and by your involuntary actions, and then you ultimately have control over this within limits because you have the external sphincter, which is voluntary muscle. So let's move into the accessory organs. We'll be talking about the salivary glands, the gallbladder, and the pancreas, and the liver. And of course, those are the four main accessory glands. Now, there are other accessory organs. Think teeth, think tongue. Those are fine, but what we want to focus on now are the actual digestive organs that are accessory, and that food does not travel through the salivary glands, through the gallbladder, through the pancreas, or the liver, but it does have a very important role in the overall digestive process. So first, of course, is the liver. Now, the liver does a lot of things. In the digestive system, we normally think of it as synthesizing and releasing bile. That bile is stored in large part in the gallbladder, but it's also storing glycogen and lipids. It's maintaining or helping to maintain your homeostasis of glucose, amino acids, fatty acids in the blood. It is synthesizing and interconverting some nutrient types so it can convert sugars to lipids and vice versa. It is synthesizing cholesterol. It is inactivating toxins, storing iron, and storing fat-soluble vitamins. So there's a huge number of digestive functions, but it's also making your plasma proteins. So think albumin, fibrinogen, clotting factors. It is also breaking down and destroying damaged and old red blood cells. That's being done by the Kupffer cells. That's a special name for the macrophages within the liver. And we're going to be storing some blood, absorbing and breaking down hormones, absorbing lipid-soluble drugs. So, oh my, love your liver. Lots and lots of things going on here. We'll get to the liver in a moment. Let me refer you back to the salivary glands. There are three pairs of glands. They're all releasing saliva into the oral cavity through some ducts. Of course, these are exocrine glands. They're releasing products into or out of the body onto an epithelial border. There is a salivary gland reflex. You know that taste buds are innervated by 7 and 9 primarily, a little bit on 10, and there's also some receptors monitored by the trigeminal nerve that are going to help with the reflex. Parasympathetic stimulation is going to increase the secretion of saliva. The three pairs include the sublingual glands. They're lying, as the name suggests, under the tongue. There are sublingual ducts that secrete into the lingual frenulum. The frenulum is the area, as you open up your mouth and you see that flap, that's the frenulum. And here you're getting a kind of a mucousy secretion. It's a buffer, it's a lubricant. It's not the big player. Only about 5% of the saliva is released from these glands. The submandibular glands are releasing through the submandibular duct, not difficult. These are releasing not only buffers, but also salivary amylase. Amylase is an important enzyme for the breakdown of starches, so starch digestion begins in the oral cavity. It's also transporting some antibodies made, and so there are immune function in your saliva in that it's helping to fight off foreign antigens. And this is your big player. About 70% of your total saliva is coming from these two submandibular glands. Finally, the parietal glands, these are the largest of the glands. They're basically deep to the skin and covering the mandible. There is a parotid duct. These are also releasing large amounts of the amylase and provide about the last fourth of the saliva. So looking at them anatomically, submandibular, sublingual, parotid gland. And then you can see the ducts that drain into the oral cavity. Now there's a number of very important things being released here. These are mixed glands in that they do have some mucus cells. They produce mucus, but they also have a serous side to them, and they're releasing enzymes like amylase and lysozyme and transporting antibodies from the interstitial fluid into the saliva. So these are mixed glands. And if you look at them under the microscope, you'll see the white non-staining glands. Those are going to be the mucus glands. Mucus, having a large fat-like component, does not stain very well, whereas the serous cells stain more darkly. Those proteins that are being made stain more darkly. Saliva is a mixture of these mucus and serous secretions, and you are, on average, producing about 1 or 1.5 liters of saliva each day, most of which is water. It's constantly flushing the oral surfaces, keeping the pH near neutral, helping prevent acid buildup that would feed the bacteria and leading to caries. It is going to be releasing those antibodies and the lysozyme to help reduce the number of bacteria in your mouth, and it's going to help lubricate the oral cavity to make it easier to form and swallow the bolus of food. The liver, let's go here. This is going to be our biggest stop, and this is your largest abdominal pelvic organ, and it is wrapped by a pretty tough fibrous capsule. It is covered by the peritoneum and has 4 lobes, right, left, caudate, and quadrate. Not difficult. Anatomically, it's going to be located, large organ here. It's there's a falciform ligament that I mentioned earlier, and you can see it is lying. Here's the stomach and the spleen, just to give you some orientation, and just a glimpse of the pancreas is shown here. The lobes, the left lobe, the right lobe, these are separated by the falciform ligament. So that's a way to tell that. There is a bare area. This is where the liver contacts the diaphragm, so quote, there's nothing going on there. And there's a small area where there's no peritoneal covering, hence its name, the bare area. There's also the round ligament, and this is going to help basically mark the path of the fetal umbilical vein. So the umbilical vein becomes essentially the round ligament. The caudate lobe, there's going to be an indentation left by the inferior vena cava, and then the quadrate is located between the left lobe and the gallbladder. Associated structures of the gallbladder, as the liver is releasing bile, it is going to release it, and it's going to be stored in the gallbladder. There is the common bile duct. This is going to carry the bile from the liver and the gallbladder, both. That's why it's called common, and down to the duodenum. And there's also a region called the porta hepatis, and this is, quote, the doorway to the liver. This is the area where most of the blood vessels and other things going into and out of the liver, along with the lesser omentum, are found, and this includes the common bile duct from the porta hepatis. Looking at the four lobes, here's the bare area on the top, the right lobe, larger left lobe, the falciform ligament, and the gallbladder sitting anteriorly here. And from the posterior side, you can see the gallbladder more clearly. Here's the inferior vena cava and the caudate lobe and the quadrate lobe, so these smaller lobes. Now, what the liver is doing, the liver is composed of some smaller lobules. We saw lobules also in the thymus as an example, just a small structure. And there are 100,000 of these lobules within the liver. And each of these is about 1 millimeter in diameter. So as I show you these pictures, keep in mind the frame, the size frame here. And each of these lobules is separated by an interlobular septum. That word makes sense. Between the lobule structures, there's a divider, a septum. Now these lobules appear as a roughly hexagonal shape, and they're going to be surrounded by 6 portal areas. Now these portal areas are more commonly referred to as the portal triad, and the triad, there's a hepatic portal vein, there's hepatic artery, and a bile duct, and these 3 are going to run side by side, and I'll show you how they are interacting within the lobules. The cells that make up the liver are called hepatocytes, and they are going to form these plates along the spokes of the wheel within these lobules. These are only 1 cell thick plates, and they are going to be covered by some microvilli. The plates are separated by sinusoids, that is big sinuses or spaces. These sinusoids are filled with delicate blood vessels. There's no basement membrane, so basically they're like fenestrated capillaries. Lots of holes, lots of interaction. The blood is being filtered and detoxified, and so we must have a lot of movement of molecules. So looking at this cartoon, we can see the hexagonal arrangement, and at each of those 6 points there is a triad. So triad, each of these, the triad is representing the green being a branch of the bile duct, the blue being a branch of the hepatic portal vein, and the red being a branch of the hepatic artery proper. So that's your portal area or your portal triad. Then there is an interlobular septum, that is the divider, if you will, that we see going around and creating this hexagonal arrangement. In the very center, there's a central canal that I'll mention here in a moment. Now what's going to happen is that, remember that blood is going to enter the liver sinusoid. So into these spaces, blood's going to come in. That blood's coming from the hepatic portal vein and the hepatic artery. So about 1/3 of the blood coming in is arterial, oxygenated. The 2/3 is coming in from the hepatic portal vein. This is deoxygenated blood and is carrying in it the nutrients that were just absorbed by the intestines. The hepatocytes that are adjacent to the sinusoids are going to be regulating the solute movement and the nutrient levels by a combination of absorption and secretion. The Kupffer cells that are in here, the macrophages, they're lining the sinusoidal areas, and they are going to be engulfing, destroying any pathogens that make it into the liver. They're going to be getting any cellular debris picked up and also helping to break down any damaged blood cells. Also, in breaking down the blood cells, they're going to be storing the iron that they get. They're going to be making and storing some lipids. They're also going to be getting rid of any heavy metals that are absorbed through the digestive tract. And then all of those sinusoids are going to drain into that central vein that was shown in the very center of the hexagonal lobules. And then eventually all that blood from the central vein drains into hepatic veins, which then drain back to the inferior vena cava. So what we've done now is we've taken nutrients from the gut, it's been absorbed and brought into the liver through the hepatic portal vein. the liver is now going to filter that blood, including any toxins, any bacteria, anything that is damaging is now going to be, quote, detoxified and then safely returned into your bloodstream by dumping back into the inferior vena cava. Now, in addition to filtering the blood, the liver and the hepatocytes are also making the bile, and the bile is going to be traveling through narrow channels called bile canaliculi. Recall we had canaliculi back in bone. The canaliculi were the small canals. And all of those canaliculi are going to merge into bile ductules and eventually into the bile duct and eventually brought out through the gallbladder and through the common bile duct to help digestion of fats. So we're looking at sort of a blow up of the triad, and what we have now is blood coming in through the hepatic portal vein branches, and the blood is traveling toward the central vein. And as that blood is traveling, it's going through these large sinusoid spaces that are surrounded by hepatocytes where there are Kupffer cells found, and through this sinusoidal travel, the cells are going to be detoxifying, taking up and destroying potentially harmful molecules, and then draining it all into the central vein. So you see all this surface area is draining toward the central vein. Now there's also the bile. These cells, let me clear this, these cells, the hepatocytes, are also making bile, and that bile is now traveling through these little tiny bile canaliculi, and the bile is actually going the other way. So bile is traveling from the cells, where I'm just kind of circling some cells here, and the bile is moving this way. Okay, so the bile is moving toward the triad. So you have bidirectional movement. You've got bile moving toward the triad, and then you have nutrients traveling from the bile duct, from the hepatic portal veins going toward the central vein. So you've got bidirectional movement of molecules, and they're all sort of mixing and blebbing together, the blood supply versus the bile supply. Now those are separated, but all the nutrients and the blood are mixing in these sinusoidal spaces. Now, if the liver is damaged, there are a lot of problems because the liver does so many important things. So if there's any kind of damage to the liver itself, constriction of blood flow to the liver, this can lead to some serious problems. Examples, viral hepatitis, a virus that causes a disease and inflammation of the liver, starts to break down the flow of and the interaction and the detoxification properties of the liver. Alcoholism also can lead to some significant changes and damage to the liver. If you have constricted blood flow or a clot going or causing some sort of problem, you can have portal hypertension. This is going to increase the pressure on the hepatic portal system. It's going to distend the veins and the capillaries and cause rupture of those. And this can be very dangerous because there's so much blood flow going through the liver that this can be a very, very dangerous hemorrhage. And also, that's going to push fluid out into the peritoneum, forming ascites, that inflammation of the abdominal cavity. The gallbladder, you've seen it before. It's that little pear-shaped organ underneath the liver, if you will. It has three sections: a fundus, the dome, the body, and the neck. And again, this is going to not only be storing but also concentrating the bile that is released from the liver. The bile is important. Bile salts are going to break down lipids and allow them to be, quote, emulsified. This is the first step necessary to carry lipids in your blood. You know, fats and water don't mix, and so we need to, quote, emulsify them, and this is going to increase the ability of them to be broken down by enzymes. The bile is going to leave the right and left hepatic ducts, and that's going to bring it from the bile ducts and form with the common hepatic duct. And then bile will join into the duodenum through the common bile duct, or there is the cystic duct. So the cystic duct is taking bile, and as it comes down toward the duodenum, it takes a turn and goes in the cystic duct and is stored and concentrated in the gallbladder. And then when you drive through the golden arches, or when your body senses a lot of fat, remember we talked about some of the hormones in the gut that sense fat and are now going to cause constriction of the gallbladder and release of bile. The bile will be released into the duodenum at the duodenal papillae, and there is a sphincter here that controls this movement called the hepatopancreatic sphincter. So you can see the three parts of the gallbladder. You can see here the right and left hepatic ducts coming down, going into the common hepatic duct. The backward flow would be into the cystic duct, and then the common bile duct dumping into the duodenum. The sphincter is controlling that release of bile, but also controlling the release of pancreatic products that we'll be discussing in a moment. There's about 1 liter of bile produced every day, and that bile will flow into the gallbladder for storage, but if the hepatopancreatic sphincter is open, then it would be going directly into the duodenum. So unless this is open, it goes to the gallbladder. If it is closed, then it's going to go back. And again, CCK, we talked about, is going to trigger that release into the duodenum. So the sphincter will dilate, the gallbladder will constrict, and bile will be dumped into the duodenum. So looking at that pathway again, CCK works on opening up the sphincter and releasing gallbladder stored bile into the duodenum for emulsification of the fats. The pancreas, very important organ, of course. We think of the pancreas primarily as being a endocrine organ, because we know about the story of insulin and glucagon and somatostatin and some of the other hormones that are released by the pancreas from the islet cells. But the pancreas also has a very important exocrine function in releasing important products into the digestive system. So the pancreas is retroperitoneal, as are the kidneys. It is extends laterally from the duodenum toward the spleen. It's a very slender organ. It is rather small in size and is divided up also into lobules and has 3 parts: the head, the tail, and the body. The pancreatic duct is going to deliver those exocrine secretions to the duodenum. The pancreatic juice is going to have water. It's going to have ions that are important for buffering. And there's about another quart or so, about a quart of pancreatic juice produced each day. There are other smaller ducts that dump products into the duodenum. So you could dump pancreatic products into the duodenum without opening up the hepatopancreatic ampulla. So there is a way for the pancreatic juices to get to the duodenum while not releasing bile from the gallbladder. The pancreatic tissue, when you look at it, is filled with acini. These are going to look a lot like salivary glands under the microscope, and they're releasing these pancreatic enzymes through exocytosis. So the pancreas is coming in close proximity to the duodenum and to the liver and the gallbladder. And we can see here that here's a blow up. These are the acinar cells. They're releasing the pancreatic products. They're coming through the duct, eventually merging into the large pancreatic duct, and can be released through the hepatopancreatic ampulla and the sphincter, but there's also other minor ducts that allow secretion directly into the duodenum without going through the hepatopancreatic sphincter. The enzymes are important for us to understand. There's an alpha amylase. This is a similar amylase that was released in the salivary glands. It is also breaking down the sugars. There's a pancreatic lipase. This is going to be helping to break down lipids, as the name suggests. There are also nucleases breaking down the nucleic acids, and proteases breaking down proteins. So all of the 4 organic groups are being broken down by pancreatic enzymes. Now, these proteolytic enzymes that are going to break down proteins are released as inactive proenzymes. If these enzymes were active in the pancreas, they would be breaking down the pancreas itself. And so we want to wait for them to be released and then for these enzymes to be activated. We talked about this before fibrinogen becoming fibrin, or pepsinogen becoming pepsin. It's the same idea. And the enzymes here are trypsin, chymotrypsin, carboxypeptidase, and elastase. And those are the most important ones. And they're made, again, as proenzymes. So this would be called trypsinogen, chymotrypsinogen, right? And then in the duodenum, they would be activated to become active trypsin, chymotrypsin, carboxypeptidase, and elastase. So the last thing I want to talk about are just some of the things that can go wrong and when there's a disorder of the digestive system. If the salivary glands are releasing or not releasing products properly, then there can be an increase in periodontal disease. This is going to lead to loss of teeth, and this is going to increase bacterial activity, gingivitis, inflammation of the gums, tooth decay, and breakdown of the periodontal ligaments that are important for maintaining the bony structure of the tooth. There can be infection of the salivary glands. For example, the mumps virus. We don't see this very much anymore because of immunizations, but the mumps virus would make a very, very large parotid gland. This could also affect the gonads and the meninges. So mumps can lead to sterility because it can damage the gonads. This is typically, in the day when more people were getting this, this would be school-aged children, but if you wait, like I said, it can cause sterility in post-adolescent males. So it's important that people get immunized so that they don't get mumps later in life and then face the possibility of sterility. Esophagitis, basically inflammation of the esophagus. This could also lead to permanent relaxation of the lower esophageal sphincter, leading to constant or often reflux. So gastroesophageal reflux, you've got the acid content of the stomach going back up into the esophagus, burning and eroding the tissues, causing what we call heartburn. Hepatitis, any sort of inflammation of the liver. This is caused by infection. We got viral infections, drugs, alcohol can all cause hepatitis. Cirrhosis, remember cero means yellow. So with a problem with the liver, there's a yellowing of the eyes, a yellowing of the skin, because there's a non-ability to break down some of the bilirubin molecules in our body. There's a number of different types of viral hepatitis, A, B, and C, some being more critical than others, but they all destroy liver cells and cause inflammation and damage to the ability of the liver to properly filter and eventually destroy the function of the liver. This can lead again to the bilirubin yellowing of the eyes and skin, a condition called jaundice. Gallstones can accumulate basically when there is high amounts of insoluble minerals and salts. They can form, and when the bile becomes too concentrated, and this can form gallstones that are not able to get through the common bile duct. And this can be a very, very painful situation, and oftentimes the gallbladder is removed in these inflamed states. So if the gallbladder gets too inflamed, again, and it blocks the cystic duct or blocks the common bile duct, very, very painful and usually results in surgical removal. Bile is still being produced by the liver, it just isn't being stored and concentrated in the gallbladder. So people after having their gallbladder removed will sometimes complain about having indigestion or difficulty breaking down a fatty meal, but overall they are doing just fine. And then gastritis, inflammation of the mucous membranes of the stomach. This can be caused by aspirin or alcohol, and this can also be caused by stress, leading to ulcer type things, bacterial infections, or even ingestion of strong chemicals. There can be peptic ulcers. This is an erosion of the stomach lining or the duodenal lining. So a gastric ulcer versus a duodenal ulcer, right? Just about the location of that ulcer. And we now know that Helicobacter pylori, a bacterium, is actually responsible for a large number of ulcers. There are antibiotics available to kill off the H. pylori, as well as drugs like Tagamet, which can inhibit acid production and decrease the problems with ulcers. Pancreatitis, inflammation of the pancreas. Again, you're going to be blocking, you could be blocking the movement through the duct. There could be an infection. There could be, again, toxicity from drugs and alcohol. And injury to the pancreatic cells can activate lysosomes, which can actually digest the pancreas itself. And if this over-digestive process does not stop, then it can be a deadly situation. So lysosomal enzymes are overproduced and destroy the pancreas in this pancreatitis. Enteritis would be an inflammation of the small intestine. Again, any number of things can cause it. There can be infection, Giardia, for example, that could cause diarrhea. And then dysentery would be diarrhea containing blood and mucus from significant inflammation. Gastroenteritis, again, inflammation of the the stomach, or the intestines due to some sort of infection, could usually be caused by poor sanitation or low quality of water. There can be colitis, inflammation of the large intestine or the colon. This would often be associated with diarrhea, or it could be with constipation, so either too much fluid or not enough fluid moving through. Constipation, of course, is going to lead to dry, hard feces and make it very difficult for defecation. There's cancer of this whole system. There can be colorectal cancer, a pretty common cancer, third most common in the U.S., affects men and women equally, and those over age 50. So starting age 50, most people will get a colonoscopy to look for these early signs of colorectal cancer. And risk factors have been identified as being a diet rich in animal fat and low in fiber. So eating your fiber is helping to minimize colon disorders and colon cancer. There are forms of inherited colon cancer, and the cancer starts off as small, localized polyps, basically benign tumors, which can become metastatic. And so during a colonoscopy, these polyps can be removed, and people with a family history should be having a colonoscopy often and having these polyps removed routinely. So that brings us to the end of the digestive system. Relatively short discussion with you. So we have about an hour and a half total on the digestive system. These are the two presentations that were not going to be discussed live. And there is a quiz and a homework assignment on mastering to help you prepare for the exam that will cover the digestive, the respiratory, and the lymphatic systems. On Thursday will be the review session and the last little bit of the respiratory system.